Hello guys, today we're going to talk about enzymes. Before we start our lesson today, I want you to subscribe to my channel and hit the notification for bell for more videos. And if you have any comments, please write them down. Before we go into enzymes and we know what the definition of enzymes, let's go and know briefly about what is digestion. Okay, digestion is the conversion of large food molecules, which are polymers, into smaller ones, which are monomers, by means of hydrolysis. This process is catalyzed by an enzymatic action. So here it's very important for our body to break down large complex food substance into simpler ones in order to be absorbed by the cell by diffusion or active transport, as the cells then use these simple compounds as a source of energy in the building of new tissue. So here for the for the digestion of examples, I'm going to tell you some examples. For example, protein is broken down into amino acids, and starch is broken down into glucose, and fat is broken down into fatty acids and glycerol. As our body cannot absorb these food like proteins just like that, it has to be broken down into very simple molecules in order to be absorbed and be used by our body. So what happens there, it's very important. This happens with the help of enzymes. So let's now know what are enzymes. They are protein substance which has the properties of a catalyst and has the ability to activate a particular chemical reaction. So here enzymes are biological catalysts and they speed up the reaction. They are not destroyed during the reaction. This means that they don't take part in the reaction. They can be used over and over again. A cell contains thousands of different types of enzymes, okay, and each enzyme is specific for each type of chemical reaction or type of food. The mechanism of enzyme action, the enzyme's ability to activate a particular chemical reaction depends on two things: the reactants and the nature of the enzyme. When the reaction is completed, what happens? The resulting molecules break away from the enzymes, leaving it in the same form as before. We have to know this is really important. We have to know what are the characteristics of enzymes. They are very specific, which means that each enzyme acts on a specific type of food. Secondly, they don't affect the products of the reaction as they work as catalysts. They only speed up the reaction, speed it up, okay, until it reaches equilibrium. Most have a reversible effect. This means they return back to its original shape. Their activity depends on temperature and pH of the medium. I'm going to tell you later why pH is the examples for pH medium. Some are secreted from the cells in an inactive form, so they need certain substance to activate them, like pepsinogen in the presence of HCl is converted into pepsin in the stomach. So, and pepsin is an active form, so now the pepsin can act on the protein and breaks down protein into amino acids. Okay, so it's very important. This process happens, for example, this digestion of protein, okay, happens in the stomach. And this is a very nice diagram here. The mechanism of enzyme action. Can you see this is the substrate? This is the substrate, and here is the enzyme. Okay, they here form enzyme substrate complex. Can you see they bind together here? And then after they break down, so the enzyme has broken down the substrate into two monomers, okay, and this is the enzyme here. Okay, let me know your comments, and if please subscribe to my channel and hit the notification bell for more videos. Thank you for listening.